Hi, everyone. Thanks for coming. So I'm happy to introduce Maya as our first intern presentator. She is a recently graduated from Morgan State University. We're really happy to have her this summer. She did such a great job. She took up an interest in filming a lot of the things that we're doing. So if you are interested in some of the things that she did, check out her YouTube page as far as Maya can see. So I will let her talk about some of the research that she did this summer. So come on up.
Thank you, Brittany. That's me. Good morning, everyone. My name is Mya Sharpe, and I'm a recent graduate of Morgan State University. And some of my project is on using PCR assay to detect dermal disease in Maryland Waterways and Eastern Oysters. So oysters are significant for many reasons. They create a source of food for humans and wildlife. They construct habitats for hundreds of small and juvenile fish, crabs, mussels, shrimp, and many other species. They have economic value as well. So according to NOAA's fisheries, the restoration of oysters can bring in a 150% increase in blue crab harvest and an additional 10 million in annual fishing revenue. And finally, they improve our water's quality. So the average adult oyster can filter up to 50 gallons of water a day. So prior to the dramatic decline in our bay, the oysters were able to filter out the entire bay, which is 19 trillion gallons in just 1 week. However, unfortunately, with the remaining oysters that we have now, it takes them about a year to do so. So the oyster populations face a variety of threats: habitat degradation, predation, mass harvesting, climate change, and disease. So the spread of disease and climate change are directly related. So since derma was first detected in the Gulf of Mexico in the 1940s, it is used to those warmer waters. So now with climate change and the increasing temperatures spreading up the East Coast, the disease has been able to spread and make its way up as far as the New England states. And it also increases the winter temperature, so it widens the window of opportunity that the disease has to infect oysters. So there's many diseases that infect oysters, but the one that remains top offender is the endoparasite Pachyderm marinus, also known as dermo, that is responsible for mass mortalities experienced by the Eastern oyster. So the way the disease is transmitted, you have an uninfected oyster, and then ingested, then it makes its way through the gills and the mantle, and then it proliferates throughout the oyster, making its way to digestive glands, and then ultimately causing tissue lysis, enters the bloodstream, causing circulation blockage, and then kills the oyster. So this disease does require the oyster to be able to reproduce. The purpose of our study. So despite the dermal prevalency, it has been reported to decline in recent years, and it remains detected in oyster populations in the Delaware Bay. And this project will help us to assess if the mortality in our oysters are caused by disease infection. So this summer we had a task of bag splitting, where we would go to the pier and retrieve the oyster bags, separate the oysters that are alive from the ones that are deceased. And during that, we noticed that significant mortality was in our oysters. So now the question is this caused by disease, or is there another factor in play? So our objectives is to investigate if there's any dermal infection in Maryland oysters, and to determine if there's any prevalency difference among different oyster lines. On to the materials and methods. We'll start off with our study region. We have the Chesapeake Bay and then the Delaware Bay. So we have 12 oysters from the Patuxent River lines C and B. The B just means a diploid line, and D is a triploid line. Then we have 10 oysters from line B in New Jersey, 12 from both the Potomac River and Eastern Shore, and they're both commercial lines, for a total of 46 oysters. So first, we shuck open the oysters, we take gill samples, and we store them in 95% ethanol. From there, this is a video of us doing the DNA extraction process. So after we take the gills, we extract the DNA from the other parts, other macromolecules that are composing the tissue, which is the proteins and the lipids. Following the DNA extraction, we must make a reaction mixture, and it is composed with these reagents here. So PCR buffer, DNTP, forward and reverse primer, Taq polymerase, and H2O. So when we get our DNA extract and our reaction mixture, we mix it together, brings us to our next step, which is PCR, polymerase chain reaction. And this is a technique that is used to make multiple copies of a segment of DNA fragment, resulting in many copies from just a small initial sample. So this is the way the PCR would work. We would take this solution now, enter it into the machine, and it will begin with double-stranded DNA. Now, with the right temperature, the DNA begins to unwind, beginning the DNA strand. So now I have a question for the audience. So by a show of hands, when I get this DNA extract, who thinks that it's just oyster DNA that I will find? Who thinks, okay. Who thinks I'll find other types of DNA as well? Okay, you're correct. So since dermal is a parasite, it does have its own DNA. So we need to be able to differentiate the DNA from each other. And we do that by using a primer, which is going to be the annealing stage. A primer is just a short nucleotide sequence that's complementary to the DNA of interest, and that's the dermal DNA. So the primer then attached, acting as a template for DNA synthesis, and then with the right temperature, 72 degrees Celsius, and the other reagent in the reaction mixture assists with the actual synthesis, bringing to the extended stage. And this process takes about 2 hours. Following that, we use a traditional method, gel electrophoresis. Now, this process allows us to separate DNA from each other based on their molecular size. So first, we take the DNA, we mix it with a DNA stain, we insert it, like I am right there, into the pores of the gel, and with the electrical charge in the apparatus, it pulls the DNA towards the positive end of the apparatus. So that's how it separates. Now, following the run of the gel, it takes about 40 minutes to an hour. We look at it under a blue light or UV light, and this is kind of what it's supposed to look like. Now, our results. So for the DNA extraction, we tried 2 different methods. We tried the Kellex method, which is a shorter protocol, inexpensive, and you can do unlimited sampling with it. And then the kit method is a longer protocol, more expensive, and limited samples. And you'd be surprised on which one that we picked. So we took the DNA extract prior to the PCR product just to see the quality of the DNA as it ran through the gel. And we noticed that the quality was way better in the kit, so we decided to move along with this method. So after running many gels, we weren't getting the desired results that we wanted to see. So this is an example right here with this gel. We had 3 samples from Eastern Shore, a positive control, and 4 negative controls. The problem here with this gel is that you see a strand with every single slot. We're supposed to see one with positive control, of course, but then none with negative control. So we realized that there's a problem that we're seeing in our gel. So we noticed that it is a primer-dimer interruption. So primer-dimer is just a byproduct of the PCR process. When instead of binding to the DNA of interest, they just bind to each other, like this one right here. And then that's the result that we're going to constantly get. And the other problem with that as well is that the primer-dimer PCR product has a 50 to 100 base pair, while the dermal has 85 base pairs. So it's hard to differentiate through the gel if it's dermal existing in there or not. So then we have to play detective a little bit to try to find solutions to exclude the primer-dimer. So first, based on what I've seen other researchers do to exclude primer-dimer, they increase the annealing temperature. We did that, that did not work. And then my mentor, Dr. Wu, he was like, maybe we should revise the primer itself. So we were going based off a literature, and this was the primer right here with the guanine adenosine right after each other. And then the source that he saw, you see the adenosine guanine. It's a very slight change, but very important. And then when we got this new primer, what did we get? We get it 2 days ago, and then we ran the gel yesterday. So this is very new results. It did work. There was a mistake in the literature with the primer. So we have our positive control band. They are light, because this is an older positive control, but we don't see any bands with negative control, and that's what we want to see. So although we weren't able to detect dermo in any of our samples this summer, through our efforts, we were able to determine the proper primer sequence to exclude the interference of the primer diamond. So for our future endeavors, we'll continue this project using the new primer. You would get a new positive control so we can have more definition through the gel and investigate the prevalence of dermo at other hatcheries and farms in the Chesapeake Bay watershed as well. These are my references. Does anybody have any questions? Oh. Far as Maya can see.
Oh, there it
For those of you on Zoom, you're welcome to either speak up now or type it in the chat and I will read it out.
If there's no questions, I guess.
Yes, Nate Cantor asks, can these methods to check for diseases be applied to other bivalves like clams or mussels or specific to oysters?
I believe it can be used for other diseases as well. Yes, I got a nod from my mentor, so that's correct.
So this is a general question, Maya, or kind of an observation/question. When you started this internship, you never worked in a hatchery, correct? You wanted to learn about it and see what goes on in a hatchery. Kind of uncertain whether it was going to be for you.
Yeah.
That's totally fine if it's not after all is said and done, but what's your kind of your like after the fact assessments, like everything you've worked on, like where are you at on like this type of work, pros and cons or whatever?
Okay, so the back is not working.
What?
Can you press the back,
No,
please?
it's not, no, it's- right now. No. That's not. Where do you want to go?
I want to wait, go forward. And then forward again to the question slide.
Yeah.
Okay, now this is a clip of us in the hatchery in one of our more busy days. I can honestly say that the first week, my lower back was hurting with a lot of bending down and doing things. But overall, throughout the experience, I think I learned more here than I've done in any other internship with all the hands-on work. And it being a small, tight-knit office, I've been able to learn from everybody there with my great mentors as well. And what's fulfilling about working in a hatchery is that you get to grow organisms, like you begin with spawning, and then you're able to grow them and just see their growth. And yesterday, Kayla was able to put one of our larvae cultures into the Mako Cove after it settled on the shell. So we've seen it through its few life stages. So it was pretty fun. I don't know if I will continue in the aquaculture, but I learned a lot from this experience.
What is the question?